Hi everyone. So today we have Padma Sundaram giving an introduction on EEG and MEG. She is an instructor here at the Martinos Center, and she's an expert in all aspects of MEG and EEG, starting from how the signals are generated to the hardware aspects and up to the analysis. So I think she's like the perfect person to give this kind of a lecture. Uh, if you have any questions during the talk, you can either raise your hands or you can post it on the Q&A box, and I'll interrupt her and ask the question, or leave it for the end if it's appropriate. So without further ado, I'll let Padma get started.
All right, thanks everybody for coming to the EEG, MEG, why and how. So, I've sort of organized the talk to have a few stories, sort of scientific stories, and then some findings that stem from these scientific stories. And then I also want to just give a feel for how we analyze EEG, MEG data, just really bird's eye view. And then I have like one slide on some new equipment we have in the MEG lab. So let me start with a little bit of history. This doesn't even move. Oh, there it is, sorry. Let's click. So the history of EEG actually starts before that of MEG. So I'm going to start a little bit more with the electrical side of things. So in the late 1700s, there was this this Italian electrophysiologist Galvani had this feeling that electricity was somehow involved in muscle nerve interactions. And so he did this experiment, which is the figure here. And it's just basically a setup in his garden. And he has on the table a dead frog. Okay, and the frog is connected by a wire to a lightning rod, and he set this up, and it was a storm was impending, and so he waited for the storm to come, and there was lightning which charged the rod and then got the muscle to twitch, so this basically showed that and then he did subsequent experiments where you could see how electricity was involved in muscle movement. Subsequently, in the 1800s, in Berlin, Hitzig and Frisch, they stimulated the invasively the cortex of a dog. And this was, fairly barbaric experiments looking from 2021, but they were dogs without any anesthesia, and they would open the skull and essentially put wires in the motor cortex, and they found that contralateral muscles would move. Then later, David Ferrier built on this work, and he actually topographically mapped motor function in a variety of animals: rabbits, cats, dogs, monkeys. And so from this, there was this understanding that that electrical stimulation could produce motor response, but it was still unknown whether there was some sort of spontaneous brain electric activity that could be recorded. So in 1875, Richard Catton actually did the first invasive recordings of brain activity, and he used a galvanometer to detect electrical signals in rabbit and monkey cortex. But all of this stuff was still invasive until Hans Berger showed up in 1929 and recorded the first human EEG by measuring electric potentials between two electrodes placed on the scalp. Now, this figure here is what was in his first recording, and was actually an EEG of his son. And this is the spontaneous EEG, and then he also recorded the heart rhythm, the ECG. And this is a synthetic 10 hertz wave that he produced from a signal generator. And then this is the alpha rhythm of his son, and with a similar synthetic wave at the bottom. Now he was had a huge paralyzing fear of failure and criticism, and was pathologically secretive. And so after he got his results, he had this feeling, perhaps a little correctly, that nobody would believe his findings. So he just sat on them for a decade and then published all of them as a series of 14 papers all at once in 1938. But in 1934, Edgar Adrian, who actually won a Nobel Prize for observing that the action potential is an all or nothing event, he reproduced Berger's findings. And these are from Adrian's paper. So he did eyes open, eyes closed, the sort of stuff people do now in EEG. And he correctly actually concluded that the alpha rhythm was likely originated from like a cluster of cells in some part of the occipital lobe. Now, subsequently in the 30s in Boston, the first auditory evoked potentials were recorded, and Hallowell and Pauline Davis, who were actually, I think, at Brigham and Women's, they made the first recordings of the human auditory evoked potentials, and they published these recordings also during wakefulness, during sleep. And one has to remember that this was long before there was computer analysis of these sorts of data, so they had to see a lot of things on single trial. In fact, the first average waveform was not even made, event-related average waveform was not even until the 60s. But once they were able to look at averaged evoked responses, there was a lot of excitement on identifying different ERP components and developing methods to record, analyze them in cognitive experiments. And now there are all kinds of auditory, visual, and somatosensory evoked potentials, and people have labeled very specific peaks and troughs, and they're usually labeled P or N to signify whether it's positive or negative, and then followed by the time in milliseconds when they're seen. So now moving to MEG, David Cohen, who is the father of MEG, is here at Martinos Center. And back in the 60s, David started constructing an elaborate magnetically shielded room. And he actually started by recording the heart signal. And this is partly because biomagnetism at that time, there was a lot of interest from the cardiology community, and the heart signal is a big one, and so it was considered easier to find since this was a really sensitive measurement. But the early measurements that David did were done with a million turn copper induction coil. And in his first paper, essentially was the only equipment he had was a shielded room that he had made and this copper coil. But it sort of opened up, sort of motivated the need for developing more sensitive recording instruments. And so with Jim Zimmerman, they developed the first practical squid sensors to detect biomagnetic fields. And that is this figures from their science paper. So where they have MEG of an alpha wave. So the eyes are closed, eyes are open, and then they have concurrent EEG. which doesn't match exactly, but it sort of shows a similar pattern. I also should mention that there was a parallel effort in Finland at the time to also develop ultra-sensitive squids, and by Oli Lunasma and many of his students and colleagues. And they at that time in the 70s, they didn't actually have the funds to construct a shielded room in the lab. And so at the peak of the Finnish winter, they would walk to the middle of a frozen lake with their subject. And then that's how they recorded the early MC magnetic fields of the heart in the middle of the frozen lake. And often, because they didn't have the shielded room, and often the subject was shivering and frozen, and so a lot of the trials had to be thrown out due to the Finnish winter. But there was, they subsequently did contribute in significant ways to the MEG instrumentation and analysis technology. So I just want to, now I just want to talk about the underlying sources of the MEG EEG signal. So here what we can see is, so this is just a schematic diagram. So this is the blue thing is a neuron, and there is an axon that is there is a synapse here which is bringing in an input to this neuron. Now, when an action potential arrives on at this synapse, it causes a change in the potential of this cell, and so this causes a slow response, and this could have an excitatory effect or an inhibitory one, and often you would see them labeled as EPSPs or IPSPs. So from the MEG and EEG are usually connected to the postsynaptic signals, and but from the point of view at the data that we are usually collecting, we usually use this model of a dipole. So I just want to explain how going from a synaptic effect, how you might get a dipole that you can understand. So here's a neuron, okay? And now in its default state, the inside of the cell is negative with respect to the outside. So it's usually like about minus 65 millivolts or so. This is an EEG signal, and that's the MEG. So right now we are not detecting anything. At the blue, there is a synapse here. Now let's say that for some action potentials arrive here, and because of this, some ions, positively charged ions from the outside of the cell, are now going inside. Now it's important to think of the cell as a kind of capacitor. Nothing goes in, nothing goes out unless you have some sort of electrochemical gradient that's driving the flow. So now that we've allowed, because of this synapse, there are now positive ions going in. And so the membrane property is now going to be locally changed. So you have basically this gradient, an electric gradient, a potential gradient along the length of this on the inside and along the length on the outside. And so what happens is that you have now a flow of current this way, and then also completing the path in the extracellular space. So now you will see something on your little mini EEG and your little mini MEG device here. Now, often in electrophysiology, people talk about current sinks and current sources, and all of these are always defined with respect to a viewer in the extracellular space. So from the perspective of this person here, here you have a current sink, the current is going in, and current is coming out. Towards him is a current source. And when you have a current sink, and so this is the plus would be a current source, minus being a current sink, you can get a dipole, a current dipole. And the current dipole is basically a separation of charge over distance. And so it's often written in units of nanoampere meters. So you will often also see in electrophysiology that people, when they talk about current densities, they do not often use units of ampere per meter square. They often use nanoamp meter per millimeter square. And that's because it's actually a current dipole moment density. And so this is kind of the fundamental currency in dealing with EEG, MEG data. Basically, we're trying to figure out from these extracranial measurements, what is the current dipole moment density inside of the brain. So I just want to highlight that current dipoles are not magnetic dipoles. Magnetic dipoles are basically a small current loop. This is basically a pair of charges that is separated by distance. So there are other types of currents. One might say, well, what about action potential? Currents due to action potentials. Those are really difficult to detect because the field, the action potential you can think of as a pair of dipoles that's moving in a pair of opposing dipoles are sort of moving together, and in the physics of it works out to be a quadrupole rather than a dipole, and that field decays as 1 over r cubed compared to the dipole fields, which decays 1 over r squared. So these are much more challenging to detect. So typically what we are dealing with is in an EEG and MEG is population activity. So it's from averaged across large numbers, ensembles of neurons. And you can get from this sort of the sort of macro current in the tissues going in a certain direction, and so you can get from the right hand rule the sense of the magnetic field. Now we will figure out now subsequently that MEG is really on, at least current MEG technology is only sensitive to radial dipoles. So for neurons that are oriented this way, which are inside of the sulcus, they are the ones that contribute to MEG. But for radial sources, we do not see an MEG signal. EEG, however, is sensitive to both radial and tangential dipoles, and so it actually sees a bit of everything. So just some numbers. Typically, the EEGs we measure in humans, like an alpha rhythm might be about 50 microvolts. Maybe in an epilepsy subject, you might see 100 to 200 microvolts during epilepsy, interictal spikes, or those sorts of events. In MEG, we are in the femtotesla range for brain signals, and in the heart, from the heart, you might see a picotesla range signal. Just to compare, the Earth's magnetic field is about a microtesla. And current densities, when we talk of current density in neurophysiology, we are always talking about current dipole moment densities. So it's always nanoamp meter per millimeter square units. It turns out, and there is a great paper by Yoshio Okara and his colleague in NeuroImage, where they collated a huge amount of data of from all kinds of measurements in a variety of animals and also in humans, where they found that the current density in brain tissue is a constant across species, and across brain regions, and it works out to be about one nanoamp meter per millimeter square. Often in MEG we talk of equivalent dipoles, and so people often say, well, they saw a 10 nanoamp meter dipole. It just means that from the point of view of the sensor, the field that you measured would be as if there were a dipole of that strength at that location inside the brain. There is some papers where people have calculated like equivalent dipoles for a single, for example, for a single pyramidal neuron, and it works out to be about 0.2 picoamp meters. But typically to detect something out on the cortex, you need about at least 50,000 synchronously firing neurons to get something detectable. So here's just some practical experimental details. So, you know, the EEG systems have become, this is an EEG recording setup. This is the kind of nowadays the kind of caps people use. Some of the caps use conductive gel. Some just are the kind of caps you can dunk into saline and just put it on the subject's head. That's just a page of EEG recording. Now, MEG measures magnetic fields with SQUID, which stands for superconducting quantum interference devices. Now, SQUID-based MEG sensors need to be, of course, are housed in a magnetically shielded room, but they also need to be cooled using liquid helium, and then they need to be tuned. And so the instrumentation is expensive. It's also important to remember that magnetic fields can originate from magnetic materials, and those need to be kept out of the MEG room. And of course, electric currents, neuronal currents. So the MEG sensors, the basic measurement that we are making is current in a coil. And since the magnetic fields are so small, in the inside of the MEG scanner, the field is amplified with a flux transformer, and the whole thing is made of a superconducting metal. So if there is a flux induced in this coil, then an equivalent one is induced in this pickup coil, and then the SQUID, this is your SQUID sensor basically. And so sometimes magnetic material is accidentally brought close to the SQUID, and the SQUID gets like a flux trap. And so often when we do MEG studies, we find some of the sensors just need a little, need to be heated to sort of remove this sort of residual flux. So there's different types of MEG sensors, and each of these sensors have different sensitivity profiles. So the basic one is the magnetometer, which is essentially just the magnetometer is essentially just a coil, and you can see that for a dipole placed here in the middle, the profile that the magnetometer will measure will be positive on one side and then negative on the other side. Now, for planar gradiometers are basically two coils that are placed next to each other, so that the result, it looks like a difference between the two of them. And then we also have axial gradiometers, where you have two coils that are essentially two magnetometers placed in series, one above the other, and the result is the difference between in the magnetic flux at that point in space. And in the MEG system at Martinos Center, we have a 306 channel system, and it has 102 magnetometers and 204 planar gradiometers. So just to recap, MEG sees only MEG sees tangential sources, and of course tilted sources, just the tangential part of the tilted source, and it doesn't see any radial sources. And EEG sees tangential sources, it sees radial sources, it sees tilted sources. And MEG does not care about conductivities of the different compartments, but EEG does. And so here you can see homogeneous EEG seen on the scalp for a homogeneous volume and with a skull and with a poorly conducting skin. So now I just want to talk a little bit about how the data are analyzed. So what we do, so this is true for MEG and EEG. What we have are measurements of either electric potentials or magnetic fields at a number of sensors on the scalp or outside the head. And we have a model of the subject's head. And then what we don't know are the source, the temporal behavior of the sources inside of the brain. So, so these here, so here we have basically we're going to just set up the solution using a distributed source framework. And so here in this giant matrix M, I will put all my measurements. And so it's, if you just take any row of this matrix, you will see exactly what I measured at that sensor. And if you take any one time point and you just plot it on the scalp, you can just see some sort of changing scalp map. Now, so this is a M is equal to GX plus E, and the X here is my unknown, it's the sources, and we're going to try to figure out that. So usually when you try to solve the MEG or EG inverse problem, you need to populate, you create some sort of distributed source framework. So you can either have the sources be on the surface, like just be on the surface of the cortex, or they can populate the volume of the brain. And based on the locations and the orientations of the sources and the location and orientation of the sensors, this geometry between the two gives you this gain matrix called the lead field matrix. And another way to think about it is that for any one any one column of this lead field matrix will tell you the field for a single dipole, unit dipole at one location, but as measured by all the sensors on outside the head. And X is my source, which we don't know, and then E is the noise. So now we start with this grid of dipoles, and you can either leave them sort of, you can vary they can have sort of free ranging orientations, or you could fix their direction to be along the normal to the cortex. Now this is an underdetermined problem because you have more sources than you have measurements. And so we need to find an optimal solution given our data. And so this is one, there's like a whole world of ways to solve this, and this is just one where you solve for it using the minimum norm estimate. So this is the smallest amplitude source that would explain our measurements. So this just to recap, this is how if you did MEG and EEG, you'd have an EEG cap on, and then we often in the MEG studies we have coils placed on the head to register the helmet with the subject. And we also acquire triangulated locations of a number of points in the subject's scalp so that we can register them with MRI. We put the subject inside of the shielded room and inside of the MEG helmet, and that's sort of a mock-up of what it might look like. Then you do your study and record your data, and then and then we analyze it for the underlying sources. So this is the rough pipeline. So you have your MRIs, and they are acquired before or after your MEG, and then you do either MEG with EEG or just EEG. And often during MEG studies, we record empty room noise because that's used to create a model of the noise for during our data processing. And then if you set up the sources to be surface sources, then when you can plot your results on like a free surfer reconstruction of the cortical surface. And if you set it up to be a volume, you can plot your results in a volume format. So just some software packages. Mostly, I mostly just use FreeSurfer and MNE Python. And sometimes EEG Lab. There's also MNEC and MNE MATLAB. So usually we run FreeSurfer on the MRIs to reconstruct the white matter, gray matter, and the inflated surfaces. And then then we need to get a boundary element model. So we get triangulations of all the interfaces between the different tissues. So we have the head geometry, and this is totally independent of the MEG data. This is just to create this sort of model of the subject's head. And then we register the MRI to the head position that was recorded during the MEG. And then we can make our source space. And so in this case, it's a surface source space in this example. So this at each of these yellow dots is a vector, and you could have the free orientation or along the normal to the cortex. And then you could, or if you don't want the surface one, you can do a volume source space. And then you can also make, so once we have the source space and we have the registered sensor, so sensors registered to the head model, we can make the gain matrix or the lead field matrix. And the noise covariance matrix can be obtained either from a baseline period of the data or from the empty room. And sometimes the noise can be, there could be instability and also have some sort of strange patterns. And so it's not a bad idea to regularize the noise covariance matrix. There's lots of stuff about this on the MNE Python website. And so once you do all this, you can get the inverse operator and then apply the inverse operator to the MEG data, which will give you a source estimate, either as a surface source like here, or as a volume source like here. There's a variety of artifacts that one should keep an eye out for: eye blinks, eye movements, muscle tension, respiration can cause slow drifts, and also the ECG artifacts due to the heart rate. And so you could use a signal space projection, SSP or ICA, to remove these artifacts. So let's see, what can we see? So there's, you can analyze the data in all kinds of ways depending on your experiment and what the question that you're trying to answer is. So just on the basic level, you can look at your data in the sensor space, which is just the raw measurements that you got, just plotted on this sort of mock-up. After we solve for the sources, you could look at the source distribution, sort of like as a function of time. You could just look at it on the inflated brain, or you could look at it in a volumetric grid. You could also look at the source amplitudes at different locations as a function of time. We could also look at kind of evoked data, that is event-related potentials or fields. Often people are interested in looking at time frequency plots to look at certain frequency bands of interest that may appear and then disappear depending on the task. Then there is also some interest in looking at sort of spatial distribution of different specific frequency ranges, alpha, beta, gamma, etc. And also you could look at just the frequency profile of your data. So MEG, EEG are both totally non-invasive. They are safe to use. They do have very good temporal resolution, and both reflect direct instantaneous neural activation from postsynaptic currents, in contrast to fMRI, which measures slow hemodynamic effects that are correlated with neural activity. Now, when we solve for the EEG, MEG sources, we can get information about current source densities of at different locations in the brain, and there have been a lot of successful efforts integrating it with fMRI or even with TMS. Many of these signals are clearly visible without group level or even individual level statistical analysis. And compared to PET or fMRI, you don't have to subtract between experimental conditions. And especially for MEG, we can model sources without any reference to conductivities inside the head. Just some things to keep in mind. For optimal recording, subjects should not move. And people often say that the inverse problem is not unique, but as long as you interpret the data with using plausible anatomical and physiological constraints, you can produce interpretable and reproducible results. And some knowledge of the underlying physics and the neurophysiology, I think, does help competent use of MEG, EEG. The detected signals, they reflect neuronal population responses, and they are mostly dominated by synchronous activation. Just a reminder that in MEG, it's superficial tangential currents more than deep radial ones. In EEG, we see both radial and tangential sources. Now, signals from thalamus, amygdala, hippocampus, those are really difficult with MEG, EEG. Even though the modern sensors, they do cover the lower part of the brain, like signals from, for example, cerebellum there's still an effort to observe them. And MEG does need a magnetically shielded room, which is expensive. And although external sources of interference can be a problem even when one is doing an EEG recording. I think this is fairly, there's some overlap here, but just to remind everybody that EEG detects electric fields and MEG detects magnetic fields. But also the current density in the cortex is approximately a constant, 1 nanoamp meter per millimeter square. And even though there are some ambiguities in the solution of the inverse problem, the results actually depend on your assumptions when you create your source model. As Mati always reminds us that different source estimation methods, they do give converging evidence as long as you interpret it correctly. And I just want to spend just one slide on the new OPM MEG system in the MEG lab. Since the squid sensors require, they need to be cooled and are at quite a distance from the scalp, about 30 millimeters. There has of late been a lot of interest in making room temperature MEG systems where the sensors can actually be placed almost directly on the scalp, and that way they conform to the subject's head. It's still early stages with they are called OPM MEG system, which stands for optically pumped magnetometer systems. They do have higher sensitivity just by virtue of being so close to the scalp, but they also have higher noise, about a factor of maybe 3 or 4 more than the squid systems. And they do have smaller dynamic range and much smaller bandwidth compared to squid. I would say the bandwidth cap tops off at about 150 hertz for the OPM MEG systems. So the question is whether we will see something new, and I think only time will tell. I think the law of Amara's law applies, that people tend to overestimate the effect in the short run, but underestimate it in the long run. And so new methods will need to be developed and to analyze this sort of new data. So we are still in the early stages. We've just started to, this is a heartbeat of a member of the MEG lab. So the system just starts to work, and we are actually seeing, biological signals. But the squid systems, the squid MEG systems in the 70s also started with the heart magnetic signals. So in a sense, we're kind of rounding back and starting all over. That's all I have for today, and thank you.
Thanks a lot, Padma. If people have questions, please type them in the Q&A box. We have one question already, so I will ask you that question. And this question is from Tepe. And he says, there are many MEG systems, NeuroMag, CTF, RICO, and different types of coils, squid, TMR, OPM. Do you think the results of source estimation would be different according to the systems or coils?
It should not be. I think if you set it up correctly, you should get converging results.
Okay. Okay. So you should get the same results no matter what system you're using. Okay. I actually have another question. Is you showed the slide of the with the action potential and the postsynaptic current. You have the slide.
Sorry. Many slides. Okay.
So.
Yeah.
Yes. So here I was a little bit confused. Do we not see the action potential because of the 1 by r cube, or because it's so fast in time that they don't add up across different neurons?
I have recently seen an action potential using MEG only for the first time recently. I have personally seen it with my eyes. With the OPM sensor, I have seen it because the sensor was so close to the sample. But in a human, you might not see it. It's just the 1 over r cubed. It's brutal to catch it.
But I'm just guessing that couldn't you like have something that activates a large area of the cortex or uses like a lot of trials, and then you could see it if it was just 1 over r cubed?
Well, it's very fast. So it's like a millisecond, it has to be, they all need to be hyper synchronized for you to see the actual shape of the action potential.
Right.
Where was this? I think I had a picture of it somewhere.
Yeah, that was basically my question. Is the 1/r cube the main effect, or is it synchronization which is more important? Also here I was confused. Why is it 100 millivolt? The
it's just
scale though.
that's the action potential. That's how big it is.
Okay. 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 Um,
Is there some other question?
yes, there are other questions. Yes. So I'm going to ask the next question, which is from Pralay Das. And he says, can you elaborate the effects of different noise covariance on source estimates?
That's more, Mainak, that's more your, but I mean, I didn't get in, so that's a good question, but it's almost like a topic of a whole other talk because it's a really involved, it's actually important.
Well, so from what I know is that you need to get the noise covariance. Well, I'll answer the question. So any kind of analysis you do, you need to do some visual inspection later on. So with the noise covariance, if your baseline has a standard deviation of 1, that means that you have a good noise covariance. So no matter what method you use, ultimately you want to look at the white-end evoked response, and if that one has a standard deviation of 1, that means that you are sort of satisfying the requirements of the math of the source estimation. So ultimately that's what you want, no matter what noise covariance method you use. Okay, there is another question from Pedro.
Well, has a follow-up question. He says, how do you choose between them for an analysis?
How do you choose between? You have to look at the white-end data. You have to look at the white-end evoked data. If that looks good, if the baseline looks good, then you pick that one. So so there is, of course, like you could have the noise, you could pick. So for instance, you have the choice between taking the baseline as the noise, or you could use the what is it called? The empty room as the noise model. Now it depends what you want to see. If you're looking at say resting state data, you don't really have a baseline, so then you cannot use that for your noise model. But if you're looking at evoked data, you can look at it. Sorry?
Like book studies, you can use your baseline from your own, pre-event segment.
Yeah, if it's epochs, if you have epoch, exactly. If you have epochs, yeah. And typically these noise measurements, these empty room measurements are like done every day in an Electra system, for instance. You do these every day. So you want to pick the noise measurement from the same day, so you have like a good, the noise hasn't changed a lot when you did the measurement. Okay. Okay, I see another question in the chat. Where is it? So Pedro asked, so how deep in the brain can we see with MEG, quote unquote, see with MEG and EEG signals? So, or is it just only, or can we see only in the cerebral cortex?
Well, there's two challenges to seeing anything deep. The first is that a lot of those sources are not, how shall I say, open field. So they may not even, like when you talk about the structure like the thalamus or one of those internal deep ones, they often have closed fields, and so there isn't actually a field outside of the structure that you could detect. But also because of the distance from the scalp, you just will not see it with MEG or EEG. It's just very difficult. Cerebellum you might be able to see because you can actually set up an electrode pattern that actually gives you some coverage. But I think the deeper sources are very difficult with MEG.
Mm-hmm. All right. I don't. Okay. I don't think I see any more questions. I think that's it. Thank you so much for your enlightening talk. The recording of this talk will be available on the Martinos YouTube channel. And in 2 weeks' time, we have a talk on funding opportunities. So please make sure that you're subscribed to the mailing list so that you receive the updates on the talk. And that's it for today. Thank you very much.
Thanks, everybody.
Thank you, Padma. All right.